I think we should start. There's a good number of participants now. Good evening, everyone. I'm Khushbu, Marketing Executive at Strand Life Sciences, and I'm delighted to welcome you all to today's webinar on single-cell RNA sequencing and advancing biomedical research. Strand is a pioneer in genomic testing in India with a strong global legacy in bioinformatics and precision medicine. Our work spans oncology, rare diseases, women's health, infectious diseases, and consumer genomics, helping individuals gain personalized insights into their genetic makeup. With that brief introduction, let's move on to the most exciting part of today's session, our speakers who will take us through how single-cell RNA sequencing is transforming biomedical research and clinical applications. Our speakers for the session are Dr. Arindam Maitra. We will be giving him a short intro later because he has not joined yet. We'll move on with our second speaker, Dr. Soumyava Basu. We welcome you, sir. He is the head Uveitis Services, LVPI Network, DBT Wellcome Trust, India Alliance Fellow, LV Prasad Eye Institute. He served as a consultant, Uveitis and Retina, and head of Uveitis Services, LV Prasad Eye Institute, Hyderabad. He's completed his medical training at University of Mumbai and Retina fellowships at Sri Ganpati Netralaya and LVPI. Undertook research sabbaticals with leading global experts at Doheny Eye Institute. Research focus is immune pathogenesis of ocular tuberculosis, especially infection, autoimmunity interactions. We welcome you, sir. And moving on, we have our Strand experts, Dr. Aribu Sudar John. She is the Associate Director, Commercial Research, Strand Life Sciences. She has several peer-reviewed publications in prestigious journals, and she serves as the reviewer for BMC Cancer, Frontiers in Genetics and Biology Methods and Protocols, has conducted, organized, participated, and delivered lectures in national workshops. She has a technical expertise in NGS technologies, single-cell and spatial genomics, proteomics, microarrays, primary cell culture, functional assays, and many more. And she's collated with numerous awards and honors. We welcome you, ma'am. Our next Strand expert is Dr. Pankaj Kumar. He serves as a senior bioinformatics engineer at Strand Life Sciences. He leads a team developing cutting-edge single-cell and spatial transcriptomics pipelines. UC Irvine and LV Prasad Institute are his overseas collaborations. He has been a postdoctoral researcher, ULB, Laboratory of Cancer Epigenetics, Belgium. He also has been a postdoctoral researcher, Karolinska Institute, Sweden, postdoctoral researcher in STEM India, doctoral researcher, CSIR IGIB India. We welcome you, sir. And our next expert is Dr. Swaraj Basu. He is also the principal bioinformatics engineer at Strand Life Sciences. He has authored over 25 publications spanning comparative genomics, genome assemblies, and clinical genetic studies with 1,000 citations, has been awarded with several research travel grants, has delivered talks and posters at many national and international conferences. Now we welcome you, sir, and I request you to please take the session forward.
Thank you. Thanks a lot, Khushbu. And yeah, glad to be part of this panel. Really looking forward to hearing from the experts. So I'll take a very short span of time of the audience to just prime them towards single cell, and then let our experts, Dr. Basu, Pankaj, Aribu, Dr. Maitra, speak about their experiences and the specific cases they have been working on. I'll share my screen. Khushbu, just let me know if it is clearly visible to everyone.
Yes, sir, it's visible. You can put it on slideshow.
Okay.
Yeah.
Someone has raised their hand for a question. I see Dari.
So I'm getting in touch with them. You can carry on.
Okay. Yeah. Yeah, my screen is fully visible to everyone, right?
Yes, Suraj.
Okay. So hi, good evening, everyone. And before I get started, means of course this is a session for single cell RNA sequencing. We'll hear about the techniques, we'll hear about the platforms, the algorithms, the specific case studies, clinical applications. But I'd like to go a little bit back and draw your attention towards something which we call the mechanism of mRNA bursting. So what you are seeing on this image, you can see there is the x-axis, which represents, let's say it represents something temporal. It represents the passage of time. And the y-axis is concentration of mRNA. And we have a blue line and we have a red line. I'll come back to it. So, but basically what this plot is showing is that mRNA expression in cells is not a constant process. When we measure something, when we measure gene expression, we don't say now the gene is present. It is not present forever. It is not present for the next 1 hour, 2 hours or so. mRNAs have their own half-lives. But in general, this pattern is bursty in nature. So it comes in bursts, it goes down, it comes in bursts, it goes down. And it is not consistent even within cells of the same cell type. So you pick a particular cell type, you pick 5 cells out of it, freeze it at any given time point. What is the probability of a gene to be 100% present in all 5 of them? We cannot always guess it out unless it is a very specific markers. The question is why this happens, and this is what this simulation shows. What they showed was they could simulate that cells often fluctuate mRNA levels even under steady conditions. And they proposed a mechanical origin of this bursting. The mechanical origin is, let's consider DNA as a coiled long wire, and it's sort of a twisted rope, and then RNA polymerase binds somewhere. Once it binds, there is binding of other enzymes, other proteins, and it leads to uncoiling, it leads to transcription. But sometimes this binding, as binding and uncoiling happens, tension builds up. And when tension builds up, the RNA polymerase stalls. So when there was no tension, transcription was freely flowing. And when there is tension, again, transcription goes down. And when the transcription goes down, since the RNA polymerase cannot move, more RNA polymerase come and keep binding, and it creates sort of a traffic jam. Now, when topoisomerases, they release the twist, suddenly the jam is released again, and we again see an increase in expression. Now, let's come back again to this image. So we have 2 lines, no mechanics and with mechanics. So they simulated 2 situations. With mechanics is considering that transcription factors bind, RNA polymerases bind, the traditional biology we know of, and how that would gravitate towards increase or decrease in mRNA expression. That is the red line. Sorry, that is the blue line. But then we also have the red line, which is accounting for binding of all these factors, uncoiling of DNA, and also this physical mechanism of DNA getting coiled, and it is creating a physical obstruction for the RNA polymerase, and that is leading to transcription stalling. And that becomes even more bursty. So now that we are setting up this premise that expression is not something constant, it keeps bursting between different cells. There's this beautiful experiment, and this was done in 2016, Nature Communications paper by Fujita et al. And what they did was they showed that even in a minimal in vitro system, where we just have a promoter, we have an oligo, probes binding to that oligo will give proof of mRNA expression. And we have a RNA polymerase on which a sort of quantum dot is there. So signals from there can also show that the RNA polymerase is binding. So a probe will show us that the mRNA is getting expressed. The signal from the RNA polymerase will show RNA polymerase is binding. And the observation was that RNA polymerases, they stall the arrest of transcription during elongation, But contrary to what the first study said, here it showed that when more RNA polymerases bind and that traffic jam is created due to DNA conformation, the binding of more RNA polymerases sort of gives a push. And that is what we see here. Suddenly, the push frees up more DNA, more RNA polymerase is binding, and that gives rise to the transcriptional burst. And they were able to measure it by measuring the expression of the mRNA with fast fish and the RNA polymerases together with the atomic force microscopy. And they showed that whenever mRNA synthesis stopped, RNA polymerase was stalled or arrested. And whenever transcription resumed, another RNA polymerase arrived. So I can't speak volumes of the beauty of this experiment because it shows a core statistical fundamental of measuring RNA expression. That is, it is bursty. It is guided by not only the dynamics of the DNA molecule itself, but by binding a third-party molecule like RNA polymerase. And the more it binds, it creates a sort of a self-feedback loop, and it promotes expression, and then it stalls again, and then it again promotes expression. So this brings us to one observation on single cells. And when we talk about single cells, there is this hypothesis beautifully postulated by this particular paper where they showed that when transcription factors, and transcription factors are kind of protein molecules, will bind to a certain area of the DNA, will promote binding of polymerases and other factors, and in general promote the downstream transcription of some other gene. So this study showed that when the transcription factor itself is transcribed, so this is the burst of expression of the transcription factor, nothing really happens. Then there is a lag, and then the transcription factor protein comes into picture after that lag. And when the transcription factor protein comes into picture, after a while, it brings into picture the expression of the target mRNA. So this is where the simplest model, where we assume that there is a direct correlation between the transcription factor expression and its target molecule, doesn't hold. It is slightly lagged correlation. So first there is a burst of transcription factor mRNA, and then there is a burst of target mRNA. Now, if we do bulk experiments, this behavior is completely lost. But when we do single cell experiments, when we measure these transcription factors in each and every cell and what are their targets, what this particular study showed is that they did the analysis in K562 cell lines. It has a master transcription factor, GATA1, well known. All its targets are well known due to CRISPR assays. Now, they saw that even though we don't have temporal observation, even though we have not measured the cells temporarily, we have just frozen them and measured them in one single cell experiment. Still, looking at the transcription factor and its expression of downstream targets shows an enrichment of correlation per cell compared to GATA1 versus any other random gene. So what is the conclusion we draw? The conclusion we draw is that even though there is a lag, that lag means that ideally we should always sequence everything at a T0, T1, and T2 to get the complete picture. We can't always do that. Even if we can't do that, measuring things at the single cell resolution still allows at a certain frozen time that certain cells would show the presence of transcription factor, a few other cells will show the presence of target mRNA. Overall, this behavior, cells show the serial behavior of transcription factor came, then the protein came, then the mRNA came, and when we have thousands of such cells, we can measure these behaviors. So this brings me to the conclusion of this mRNA expression bursts, that let's say we are measuring things at a single cell level, and we have a sample 1 where we are measuring a cell type 1. We generally don't know the cell types upfront. Let's assume in this case, the hypothesis, we know what it is. And then we also have a cell type 2 in the sample 1 and 3 cells in each. Then we have a sample 2 where the cell type 1 is also there and the cell type 2 is also there. At the individual level, these bars, the red and blue bars, show the amount of expression of a certain marker gene. If we had measured this in bulk, we would not get any difference maybe across the markers. But when we are looking at this marker at the single cell level, we can at least make these guesses or look at the matrix, do downstream statistics. I'm saying even if we don't use any sophisticated methodology, even if we just go with simple statistical probability distributions, we'll still know that for this cell type, overall expression is lower, here it's higher, here it becomes higher, here it becomes lower, and so on. And that's where mRNA transcription, it occurs in stochastic bursts in individual cells, and averaged across thousands of cells, these bursts get canceled out. While if we measure it at the single cell level, we can get a far better signal and understand the mechanisms better. No better explanation of this behavior than cancer. I'm not going to dissect this image. I wouldn't remember all these cell types myself. Maybe Pankaj knows, or Dr. Basu or Dr. Maitra Aribu. But what I can say is that due to single cell experiments, we have understood hidden cellular diversities to a large extent, tumor-associated macrophages. The classical model was tumor-associated macrophages either have a pro-inflammatory or an anti-inflammatory kind of a situation. But now with single cell, we know that the solution is not so easy, or the mechanism is not so simple. There is a full spectrum of states tumor-associated macrophages exist in. And so many other immune cell types, their proliferation, their behavior with tumor progression, we are able to understand. Which is where this particular study is something I found very interesting, because they looked into head and neck cancer progression. And what they saw was they measured or did single cell experiments from tissues derived from normal, pre, early, advanced, recurring, and then metastasized samples. And one clear observation was that post-TN positive fibroblasts and SPP1 positive macrophages, they play a major role in lineaging of the tumor microenvironment as the tumor state progresses, which you can see here, post-TN fibroblast expression as the tumor state progresses, and the SPP1 macrophage. And later the markers they identified from the single cell experiments, various cell types, when they took those markers and built sort of a survival model, took it back to bulk RNA-seq in the cancer genome atlas, that survival model was able to show that patients where these gene expressions are strong generally gravitate towards malignancy recurrence compared to their patients where it is not. So it's one clear example of how an exploration and discovery takes us more closer towards one step towards translation. So I'll not speak much on this. I'll just slightly deviate from the topic and take us to one more application of single cell, which is slightly outside the clinic, but still very, very pertinent regarding the life around us, which is agriculture. Why so? Because single cell is not just limited to understanding tumor microenvironments. It is limited to understanding all kinds of microenvironments, cellular heterogeneity. And this is what this particular study showed, that when they took naturally growing cotton plants versus plants which were given salt stress, and by salt stress, they mean they were grown in solutions of NaCl, they saw that these two particular cell types, which are internal in the roots, which is 12, this light blue cortex cell, and 10 endodermis cells, as salt stress is given, these two cell types start decreasing in population, while the external cell types, they tend to increase in population. And with this, they were also able to identify one specific marker present in, I think, cortex cells, or which is an auxin responsive gene. And this marker kept showing up when there was salt tolerance, and its silencing causes salt and oxidative aging. So this also shows the value of the spectrum of things we can measure, whether it's environmental samples, agriculture, plants, and of course the entire tumor microenvironment and the immune microenvironment, understanding better from the clinical perspective. So this is where I'd like to stop. I hope I was brief as I promised, and I'd like to hand over to our speakers for today. Thanks, Khushbu.
Thank you, sir. That was a wonderful presentation. Audience, I would please request you to post your queries and we will be addressing it at the end of all the presentations. Now, I request Dr. Arindam to please present his presentation.
Okay. So first of all, as I'm sharing my screen, are you able to hear me?
Yes, sir.
Yes.
Perfect. So as I am sharing my screen, I really thank Strand and Khushbu and others who are involved in organizing this to actually host me and invite me to do this. I mean, I really thank you for that. So is my are my slides visible?
Yes.
Let me just 1 sec. Yeah, perfect. So thank you for this. And I was listening to Dr. Basu's talk with interest. I had slipped into towards the end, and it was very, very interesting. So I will. So he was talking about quite a few things, and then in cancer, he probably gave a placeholder. So I will be talking to you about today about how this wonderful technology that has burst over the scenes a few quite some time back, but it has been evolving in the process, and how that has been changing the way we understand various problems in life, various problems in biology, various problems in biomedical sciences. So I'll be talking to you today about oral cancer. I need not talk much about oral cancer because everybody knows about oral cancer, what is its importance, particularly in the Indian subcontinent, and the fact that the Indian subcontinent variant of the oral cancer is actually in the site specific. It's more in the gingivobuccal region compared to floor of tongue or mouth, floor of mouth or tongue, although the tongue cancers has been on the increase recently. And it's primarily this site specificity is related to the habit of chewing tobacco, which has a very high incidence of chewing tobacco from early age in the population in the subcontinent. But what is not so well known is another associated condition with oral cancer. It's a pre-cancer condition that we can call it. It is oral submucous fibrosis. So this oral submucous fibrosis is a chronic premalignant disorder where there is a lot of inflammation and progressive fibrosis in the mouth, related to the same causes that are also related to oral cancer, which is chewing of tobacco along with primarily areca nut and a few other things. And the prevalence of this oral submucous fibrosis is very high in India. It's about more than 6%. And it's one of the highest incident premalignant disorders of the oral cavity. And there is a little variable but quite substantial malignant transformation rate, which is like 7 to 13%. So, which means that about 7 to 13% of individuals who develop oral submucous fibrosis or OSMF can also go on and develop frank oral cancer. So that is where we came in, and we in some of our earlier studies, we got some indications that the oral tumors which develop in the backdrop of this oral submucous fibrosis appeared to be behaving a little differently from the tumors that generate without such a premalignant condition. So today I'll be talking to you about using these kind of approaches to understand, to get information which no one has seen before in not only oral cancer, but the oral cancer also associated with oral submucous fibrosis. So we had been, as a group, we had been working on oral tumors quite a bit. And that has been primarily with the International Cancer Genome Consortium project, where for the last decade, more than a decade, we had been working on oral cancer and understanding the genomic landscape, genomic, epigenomic, and transcriptomic landscape of oral cancer. We have a very interesting findings, and some of these actually not only are published, but now we are trying to translate them to use for treatment or prediction of risk of oral tumors. What was interesting was when we did get a lot of plethora of information, but then invariably in these situations, what we do is we take a biopsy of the tumor tissue, or rather the clinician takes a biopsy of the tumor tissue. And that biopsy contains millions of cells. And as Dr. Basu was explaining earlier, that when we did this kind of bulk transcriptome approaches, we actually were averaging, and that tumor biopsy contains many different kinds of cells. But what readout we are getting in terms of the RNA-seq data was kind of an average of those over all those different cell types and individual cells. So the next thing that we wanted to do was to go deeper and understand how the individual transcriptomes vary. So there's a very common example that I give that in terms of bulk transcriptome and single cell transcriptome. So if you consider the tumor ecosystem of an oral tumor, it contains various diverse cells, right? It contains various different cell types which might be associated with tumor growth, drug resistance, metastasis. There are immune cells, malignant cells, stromal cells, and all these cells together represent the tumor ecosystem. Just exactly like how orchestra plays. Like in a typical orchestra which plays music, you have about 50 players playing different kinds of instruments. But you do not get to hear what individual players own music on that particular instrument that he or she is playing is. What you hear from the orchestra is the overall symphony, or what you hear is the overall raga that is being played out. And exactly bulk transcriptome is like that. So when you use analyze and try to infer bulk transcriptome data, it is like that overall symphony that we are getting from the orchestra of the different cells that are present in the tumor ecosystem. But if we were to able to deconvolute that symphony to get individual music from each of these players, we would understand how each of them contributes to the main symphony that we are hearing. And similarly, same for the tumor ecosystem, that what we do actually in single cell RNA sequencing is try to deconvolute or try to from the sequence data, we are able to actually understand how each of these cells for each cell type or cell state are expressing their genes, those 20,000 genes or a subset of those 20,000 genes, and how is that contributing to the overall tumor phenotype. Now, when we talk of cancer, we also know that no matter what cancer, there is a huge heterogeneity of clinical outcomes. And what we suspect, and now it is coming to light, is that this clue to this heterogeneity of clinical outcome lie in this entire milieu of gene expression that these cell types and cell states are contributing to in the tumor ecosystem. So that brings me to the topic that we had been working on bulk, and now we have moved on to single cells and trying to understand, gain insights in the OSCC tumor ecosystem. It's been a study that has been going on for quite some time. We have a team, a very large team, which is more represents like the periodic table of elements in a group, where as you go down, seniority goes down, increases. But again, the topmost players of this team are actually young people, of the young PhDs and postdocs in my lab who actually have driven this research forward. So although we have many contributions and we have many funders and we have many big teams supporting us and big organizations, big people supporting us, but these are the people who have actually played the lead role in getting us to where we are in terms of this single cell transcriptomics of oral cancer. So this study was conducted in Kolkata. So we wanted to take on treatment naive early stage tumor patients of oral. And for that, we conducted the largest dental college, government dental college in hospital, Kolkata. It is called the Dr. R. Ahmed Dental College, and recruited patients from there. And the resected tissues were transported to NIBMG. Thanks, and because NIBMG's location has a very direct suburban railway service with this dental hospital where it is located, we actually use the Indian Railways to give a fast transport of these samples to the institute, where we process them using a 10x Genomics Chromium platform, and then we analyze the data. And so that's what we are. We did various kind of quality controls, of course, which are standard in terms of single cell RNA-seq. Like we remove, so in the process, we removed red blood cells, we removed duplicates, cells which are expressing very low or very high number of genes. And we also, the doublets were removed, and also we removed apoptotic cells, cells where there is high expression of mitochondrial genes. So having done that, we ultimately retained 27 tumor samples from 27 patients, which is one tumor per patient, and which collectively contributed about a little more than 150,000 cells that we actually analyzed and inferred our findings. So we find various major cell types, which are like mostly these malignant cells. We identify epithelial cells in this kind of analysis, but then subsequently we developed a method, analytical method, by which we could actually identify malignant cells from these epithelial cells, because there are some epithelial cells which can be non-malignant in nature. Then we T cells, and then various other endothelial cells, fibroblasts, myeloids, B cells we identified. Now we also saw in the second UMAP plot, and I am sure by now you have realized what these plots are, that each dot represents a cell, two dots are close together if you have close expression of genes, they are different if they have different expression of genes. So, and each cluster, we identify what cell type by actually looking at the existing biological knowledge of what kind of cell expression, cell marker expressions happen in a particular cell type. So we also found that we had two kinds of patients. One who had developed oral cancer directly, and the other where their oral cancer had developed in a background of oral submucous fibrosis. When we looked at these clusters, we found all these clusters were contributed from both patients, which means that these major cell types were present in both the patients. And then we did further analysis and divided these cell types, further classified these cell types into what are known as cell states. Like the malignant cells themselves, we actually identified various cell states of these malignant cells, which I will tell a little bit later. And then we had like other cell types, we had these various different cell states of T cells. We had this transitional memory B cells. Then we divided many of these cells into various other cell states. So in total, we actually identified 43 distinct cell states from 9 different cell types in the oral tumors. We found that T cells are the most abundant cell types in the oral tumor ecosystem, which was followed by myeloid cells. But then this is nothing new, that's known by and large from histopathology data or as well, that there is a lot of T cell and immune cell infiltration in the oral tumors. They are supposed to be hot tumors. Now, if we focus on the malignant cells, we then subcluster these malignant cells and identified about 10 different subclusters. Overall, these broadly we found are explained by 4 different major clusters. So these 4 different major clusters, we call them as MAL1, MAL2, MAL3, and MAL4. So these are, and then what we could do was look at the differential abundance of these different subclusters of malignant cells between the two group of patients we had. We found that whereas MAL1 and MAL3 are enriched in both patient groups. So you can see that on this side, there is one patient group, on this side, there is other, and they are both actually represented. MAL1 is here and MAL3 is here. They're represented in both patient groups. MAL2 and MAL4 were differentially present mostly in OSMF associated OSCC. Now, just keep that in mind. We'll go back, we'll come next, we'll look into what these different 4 groups of malignant cells are. So when we did a deeper dive and tried to understand what are the differentially expressed genes in each of these subclusters or groups, states of malignant cells, we found that MAL1 is actually those malignant cells which we are very metabolically active. They express immunoglobulin genes. Now, these is basically epithelial cells which have become malignant, but they are expressing immunoglobulin genes, and they have a lot of pro-tumorigenic effect by promoting tumor growth and immune escape. So all those kind of gene expression programs are present in this group. So if you go back, then MAL3 was the other that was expressed in both all the patients. So MAL3 consists mostly of oncofetally reprogrammed. So these are cells which have started to express genes which are known to be expressed in fetal cells. So there are certain genes which are expressed in the fetal state, but then subsequently during lifetime, after the fetal state is over, those genes get shut down. But here we are finding they are actually going back and starting to express some of these fetal genes. So we call this oncofetal reprogramming. And we found that most of these were growth promoting and immunosuppressive in nature. Then we had this MAL2 and MAL4. Now, MAL2 was found to express something known as partial EMT. We know that in cancer, there is something called epithelial to mesenchymal transition, right? So the malignant cells, which are basically epithelial, start expressing mesenchymal markers, and they move out and actually cause metastasis, locoregional metastasis. So, but these cells are in the process of transition. They are not fully mesenchymal yet. They are no longer epithelial, but they are somewhere between epithelial and mesenchymal. This is known as partial EMT. And this partial EMT phenomenon has been very much associated with incidence of metastasis. So they are responsible. So the gene expression programs here were found to be more involved in invasion, collective migration, locoregional and distant metastasis. Now, MAL4, the other one, so this MAL2 and MAL4 are mostly expressed in OSMF-associated OSCC. So we found that MAL4 were again another subclass of oncofetally reprogrammed cells. But here, these MAL4 were those oncofetally reprogrammed cells which are expressing more immunomodulatory pathways and cell death signaling. So which means there is a lot more immune activity associated with these MAL4 cells. What exactly do that mean? We are right now conducting experiments downstream to understand how that impacts on the overall tumor. So these are the 4 different malignant cells. We also, in single cell transcriptome, there is an advantage that because we are sequencing multiple cells, we actually can capture many cells in different states. So if a cell is evolving from one state to the other, if I have sample cells sufficiently, I should get all cells belonging to all the intervening positions. So similarly here, if you see, so this is one cell state to which the cells are evolving to the other cell state. Actually, from the single cell data, I can reconstruct this trajectory, and although it's not in real time, we call it pseudo time. So these are like trajectory of evolution of malignant cells here. And we found that actually what is happening is the malignant 4 is probably the early stage of malignant cells, which are evolving to malignant 1, 3, and one of them is offshooting to malignant 2. So this is a typical trajectory of malignant cells where these oncofetally reprogrammed malignant 4 cells are actually evolving into cells which have starting to express PEMT, cells which are getting to express immunoglobulin genes and are metabolically active, and cells which are again a different kind of oncofetal reprogramming where there is more growth promotion and immunosuppressive expression programs. So this is what we saw in malignant cells. Then we can look at cell-cell interaction. Like if we know that there is one particular ligand in one cell and a receptor in another cell, then we know, looking at the expression of the ligand in the one cell and its receptor in the other cell, we can look at cell-cell interactions. So we looked into these various cell types. We tried to understand how these malignant cells are communicating by looking at this receptor ligand kind of analysis. And we found that in the MAL1 signatures, which is metabolically active cell, malignant cells, there is an upregulation of MIF signaling. MIF signaling is like macrophage inhibitory, migration inhibitory factor, and that is associated with immune suppression from pro-inflammatory to anti-inflammatory macrophage polarization and T cell exhaustion, which shows that these MAL1 are actively contributing to an creation of an immunosuppressive environment, microenvironment in the tumor ecosystem. Whereas the other cell type like MAL2 were more participating in cell-cell interactions which are related to ECM remodeling. So it is like they are more related to cell adhesion, invasion, and so that's also another key driver of OSCC progression. So we also then what we did was we had this TCGA data, which is bulk transcriptome data and which has a lot of follow-up information. Similarly, we had our ICGC oral cancer data, which we ourselves generated with ACTRE, and we had follow-up information. And we had looked at RNA-seq of tumor and paired adjacent normal tissues. So we deconvoluted our bulk RNA-seq data to using the signatures that we obtained for these particular malignant subsets. So we looked at MAL1 and found that actually in both TCG and ICGC data, this malignant 1 signatures expression is related to poor prognosis. And we also find that this MAL1 signature is present significantly more in the tumors compared to the paired adjacent normal tissue. Similarly, for MAL2 also, we had similar association. We found that MAL2 expressions are in TCG and ICGC are associated with poor prognosis, and MAL2 signatures are also upregulated in tumor compared to paired adjacent normal tissue. So we have very high hopes that these signatures can actually be used in not only to understand what is going on in the tumor ecosystem, but also to manage better use for prognosis and to understand how patients will fare based on the gene expression signatures. Excuse me. So we now move on to something called as non-malignant cells. We understand that I remember that I had talked about T cells. Sorry, T cells were the most abundant cells in the tumor ecosystem. Now we did a deep dive on the T cells and found that the T cells were again had different cell states like Tregs and then CD8 plus effector cells, naive T cells, natural killer T cells, and so forth. Interestingly, we identified a cell state of T cells which were found to not have expression of CD4 and CD8 both. So these are not unknown in immunology, and but they are very not so much investigated in cancer per se, and definitely not in oral cancer. So we termed these are termed as double negative T cells. So this was a group, we had substantial presence of double negative T cells in these T cells, and we found that they are mostly the most T cell states which are associated with abundant in the OSMF associated OSCC were mostly associated with cytotoxic activity, and the double negative T cells were predominantly present in only OSCC and not present much in the OSMF associated OSCC, which shows that there's a fundamental difference between these two tumor subtypes. So OSCC is the OSMF associated OSCC is more associated with immune active kind of environment. The DNT cells we again subclustered, and we found that it subclusters into 3 different clusters, and there is a gene that's expression actually explains these subclusters, which is PLCG2, phospholipase C gamma 2, and so there are cells, DNT cells, which have low expression, medium expression, and high expression of PLC gamma 2. Now, when we did a trajectory analysis in pseudo time, we found that probably the PLCG2 low cells are actually evolving into PLCG2 high cells in the tumor ecosystem. And correlated with this evolution is a decrease in T effector functions, also associated with an increased average calcium signaling. So and again we did an analysis with the TCGA cohort and found that these double negative T cells were signature was associated with poor prognosis. So what we propose, we also saw that this upregulation of PLCG2 expression is associated with genes whose expressions like BTG2. Now, these genes have been previously reported to be associated with T cell quiescence. So we have come up with a model and we are trying to prove it now downstream using cellular experiments, where we propose is that what happens is these T cells enter, lose CD4, CD8 expression, and then in presence of the tumor calcium rich environment, they start expressing PLCG2, and ultimately that leads to quiescence of these T cells. So although there are a lot of T cells in the oral tumor, but if they become quiescent, they are no longer able to take part in the immune response against the tumor cells. And then subsequently, we have shown using other methods that these double negative PLCG2 high double negative T cells are actually a reality, and it's just not a figment of our analysis. The last one that I'll talk to you about is the macrophages. We found in our study that the macrophages are evolving from M1 to M2. So M1 are pro-inflammatory macrophages and M2 are anti-inflammatory macrophages. So what's happening is the pro-inflammatory macrophages are getting into an anti-inflammatory state in the tumor ecosystem, resulting in loss of action against the malignant cells. And what's more, there are a lot of these intermediate states, like M1, M2 is a broad, and then we find that the M1, M2, which is more like M1, and M1, M2 states which are more like M2. And these, again, we found significant association with disease-free survival in the TCGA data when you use these M2 signatures. And high M1 proportions and M1 gene signatures, which are like pro-inflammatory macrophages, are associated with good prognosis in tumor patients in oral tumor patients in TCGA data. So this is what we found, that there is an extensive transcriptional diversity at cellular level in OSCC. The cellular composition and abundance of these cells, malignant cell states, have specific adaptations to the tumor ecosystem, and there are 4 major signatures, and then they partially empty oncovital reprogramming and IGKC expression, and the malignant 1 and 2 expression, at least 2 gene expression signatures we have identified, which might affect prognosis and have potential for use in future. We also found that there is an immune suppression program in the oral tumor ecosystem, and this presence of double negative T cells and macrophages in transit actually show that the tumors are getting more immune inactive as the tumor progresses. So maybe that's the one reason why there is not much immune response to the malignant cells, although there are immune cells sitting inside the tumor ecosystem. And then there are multiple distinct cell states of oncofetally reprogrammed malignant cells. That is another area that we are highly interested in, which might have important roles in tumorigenesis, progression, and therapy. And finally, we should have now a lot of information from our data that this OSMF-associated OSCC tumors exhibit distinct profiles of expression from tumors which are not associated with OSMF. So I end here. And basically, this is my whole lab. We work on, in addition to oral cancer, we also are investigating preterm birth, and we are using single-cell transcriptomics of placenta to understand preterm birth. And we have also participated in a large initiative called the Asian Immune Diversity Atlas, where we have profiled using single-cell transcriptome and ATAC-seq peripheral blood immune cells of normal individuals. And from there, we are trying to correlate with what might happen if there is, in cases of disease severity with RNA virus infections. So the particular group which most significantly contributed here in the study I presented, he is our clinician collaborator, Dr. Sandeep Ghosh, a very excited clinician, and who is probably as excited, if not more excited than us, and many times comes up with a lot of interesting questions. So PhD students and postdocs at various levels, particularly Arunima is singled out. She is in her 5th year and now she has discovered some of these in the larger data set and now trying to come up with cellular models and cell-based mechanistic inferences on some of the observations we have seen. And then Arpita and Samriddhi are the new students who have joined. Samriddhi is working on colorectal cancer and a different cancer type, but trying to see similar observations which might be happening there as well. Also have a very large technical group, which is our genomics lab, genomics code facility, which I also run, and it's a very exciting place out there. Thank you very much. I think I'm done. So any questions, I would be happy to take.
Thank you, Dr. Maitra. That was a very insightful presentation. We do have questions, but let us take up at the end because Dr. Soumya Basu's presentation is next. So, sir, I request you to please take the session forward. It's visible, sir.
Yeah. Okay. So thanks for including me in this presentation. And I'll be talking about single-cell RNA-seq from ocular samples, specifically the vitreous fluid taken from eyes with inflammation. So the inflammation in the eye is called as uveitis. So we are looking at chronic uveitis in the eye. And I've given a very tacky subtitle called Tackling One Cell at a Time. I'm sure you would have heard this before as well. So we have already heard two very elegant presentations and very detailed presentations on the principles and then the application of single-cell RNA-seq. In that sense, my presentation is more of an overview, partly because all of this is unpublished, and I'm not going too much into the characterization of the individual clusters that we got from our samples. So I'll start with the story of this gentleman, Alex Shalek. His name was there in the, amongst the authors of the previous presentation. So when he was a postdoc fellow at MIT in 2013, and he was working with dendritic cell cultures, he found that he was not getting reproducible results in his experiments, and he was quite frustrated because he thought that his technique was quite sound. So he began sequencing each cell individually, and that is when he saw that each of the cells, while the housekeeping genes were expressing pretty normally in all the cells, the variable genes, which were the immune response genes, had extremely variable expression between different cells. This can also be seen in this RNA fish image, where you can see the IL-6 expression is so variable between different cells, same with the CXCL1. So the bottom line is cells that look identical need not be alike. They are very heterogeneous, and this was the birth of single-cell RNA-seq. So from this time when these 18 cells were described, we have moved on to a stage where I'm going to talk about the first stage of our single cell data. We have a lot more data that came up after this, but these are 21,195 cells that we got from the vitreous samples of human uveitis, and we can see that we are getting almost 20 clusters of immune cells from these uveitis samples. So why do we need to look at these cells individually in the context of ocular inflammation or uveitis? This is because chronic inflammation is the most common reason for all the structural complications that happen in uveitis, like cataract, glaucoma, etc. Cause of the vision loss, cause of a lot of drug toxicity, and such long-term treatment also leads to a lot of psychosocial impact. Now, this can be accounted for by the cellular heterogeneity, which could manifest in the gene expression, metabolic programming, or the signaling pathways, and which could be the reason why one treatment does not fit all. Now, when there is treatment failure, it leads to further chronic inflammation and cellular heterogeneity, and this vicious cycle goes on. So to break this cycle, we need to understand the heterogeneity in our samples and plan our treatment accordingly. Now, there have been quite a few publications on the single-cell immune landscape in human uveitis, both from the front of the eye, which is called as the aqueous humor, as well as the back of the eye, which is called as the vitreous humor. Now, you can see there is a full spectrum with some types of diseases like endophthalmitis or acute anterior uveitis having predominantly the innate type of cells, whereas the other types like the granulomatous uveitis or the Vogt-Koyanagi-Hara disease having predominantly the adaptive immune cells, especially the T cells. And our area of interest, the type of samples that we get for our research are mainly from this group of idiopathic granulomatous uveitis. The question we asked was how heterogeneous is the vitreous immune cell population in chronic uveitis, and if there are any novel, rare, or transient cell populations that we can associate with chronic inflammation. We selected patients who had endogenous uveitis, mainly in the back of the eye, and had particular classification as acute or chronic. And these were, and we excluded patients who had a known clinical pattern, because these are the patients who will not need a surgery to get any samples. So these patients are not part of our study. We had 4 patients in the initial cohort, and these are listed here. Specifically, you see that the range of the duration of disease is extremely variable, with patient number 2 having just 1 month of disease, whereas patient number 3 had 30 years of follow-up in our institute. 2 of these patients, number 3 and 4, also had some evidence of tuberculosis, which is a common cause of inflammation in the eye, at least in TB endemic countries. So broadly, the protocol we followed that in our institute, which is LV Prasad Eye Institute, we collected the samples by a surgery called as pars plana vitrectomy. These were collected in the RPMI medium. And the cells were transferred to CCMB, where on the 10x genomic platform, of course, the vitreous cells were isolated, and there was a parallel RNA and TCR 5' sequencing, 5' profiling that was done. So after the libraries were constructed, these were then moved to Strand, and that's how we got connected with Strand Life Sciences, where all the data analysis was performed. And Pankaj, who is speaking later today, he and his team has done all the data analysis that I'm going to talk about. So we are already familiar with this 10x Chromium single cell platform, and all the various methods that we use for preparing the samples. So as I mentioned earlier, this was a 5' gene expression profiling. Now, this is the output that we got from the cell ranger for sample 1. There are about 5,200 cells with 22,300 mean reads per cell and about 2,000 median genes per cell. This is the quality metrics that you are seeing here, the encount RNA, the feature RNA. But what made us most happy was the very low mitochondrial RNA that you see here, because this was a major issue when we started off. It took us several months of hit and trial to remove the dead cells from our sample and get such high quality data for our analysis. So at the end of all of this, we had variable number of cells from each sample, with sample 3 having the least number of cells, about 3,100, and sample 4 having nearly 8,000 cells, as can be seen here. So together, these accounted for 21,195 cells, which were pooled in for this in this UMAP, and at a cluster resolution of 0.6, we have about 20 clusters that you can see here. Now, most of these are the T cell clusters, CD8 and CD4 T cells. The ones that you see below, these are mainly the myeloid cells. On top, cluster number 12 is the gamma delta T cells. Cluster 18 is the very small population of B cells. Cluster 6 is the NK T cells, and cluster 7, which you see here, are the T regulatory cells. So as I told earlier, the green and the orange that you see here, those are largely the CD4 and CD8 T cells, and they accounted for the majority of cells in our samples, all of which came from different forms of granulomatous uveitis. You see the other cell types as well. Now, as is very elementary in single cell RNA-seq, the distribution of these individual cell types was extremely variable. Interestingly, you see that in the sample 1, which is the lowest UV1, you see that the CD8 population is relatively high. Similarly, in sample 3, you see that the myeloid cell population is relatively high. So this was the essence of doing this single cell RNA-seq, to show the variability in the distribution of individual cell types. If we look at the CD4 cells alone, these are the CD4 clusters, and you see, I know the print is very small, but there are the large green one is the naive-like cells, and there are a lot of cytotoxic CD4 cells that you can see here, especially cluster 1. Keep that in mind. This is cluster 1, which is a cytotoxic CD4 cell. When you look at the clonal distribution, you see that expectedly the non-expanded ones are mainly in the naive cluster, whereas the expanded ones are occupying the other clusters. We have looked at the different clusters separately in each of the 4 samples, and just to get a sense of it, you see that the cluster number 7 is very highly abundant in sample 3, whereas it is nearly absent in sample 1. So this shows the identification of, and remember, cluster 3 is the one which had a 30-year disease. So, this might be one of the clusters that has a role in really chronic inflammation. You also see the expansion profile. So this cluster 7 in sample number 3 also has a very high abundance of hyper expanded and large expanded cells, as can be seen by the yellow and the orange color. Now, the other thing which we already heard about it is the RNA velocity analysis, and here it is done using the scVelo algorithm. This mainly looks at the splicing of the RNA transcript. So if there is more of unspliced RNA, then it shows that those genes are upregulated, and if it is more of the spliced RNA, it shows that these are the terminally differentiated cells, and here the genes are getting down-regulated. So this is the essence of the scVelo analysis. You see this picture here, which is used to demonstrate the different states where you pick up of this woman jumping from the rock into the sea. And if you combine these different states, you know where it was at a given point of time. So that's how we, what we plan to gain from the RNA velocity analysis. As you can see here, cluster 1 is, the cytotoxic CD4 cell where most of the arrows are pointing. If you look at the CD8 plus T cells, you see that there are 8 CD8 clusters. Again, the top ones that you see here, cluster 1 especially, these are the exhausted CD8 cells. There is also a mate cell cluster, which is here on the cluster 1 on the left. And then there is a CD8 TM cell. So you see the clonal distribution, which is quite evenly distributed. The expanded cells are across all the clusters. But again, just like the CD4 cells, you see that if you look at this particular cluster 3, it is the highest in sample number 2, which, if you remember, was the one with acute inflammation, just 1 month history. So this CD8 cluster is highest in sample 2, whereas it is the lowest in sample 3, which had the 30-year history. So, this again gives a sense that the duration, the chronicity of the inflammation could influence the distribution of different clusters, T cell clusters, in the vitreous samples of these patients. Again, you see the SCVelo analysis for the CD8 clusters, and you can see that the arrows are pointing from cluster 0 towards cluster 1, which is the exhausted CD8 cluster. So why do we need to know all of this? I think we have still not started really applying our knowledge, the very high resolution knowledge. You heard so much of it in the previous talk that we get from our single cell data. But one of the case reports that was published on a patient with drug-induced hypersensitivity syndrome, which is also called as DRESS, because there is eosinophilia in these patients, to the drug called cotrimoxazole, which was being treated with many types of immunosuppressive therapy, and none was working, which is when the team went for this single cell analysis of the blood samples. And that's where they saw enrichment of the JAK-STAT pathways. You can see all the markers here for the JAK-STAT pathway. And not only that, they also found that there was the HHV6B DNA in the CD4 TCM cells. So this also, the viral infection was also noted on the single cell libraries. And from here, the patient was treated with a JAK inhibitor, which is tofacitinib, and antiviral valganciclovir, leading to finally resolution of the inflammation in this patient. Now, this is a relatively old paper, 5 years old. There are several other examples now where single cell analysis has helped in precision treatment of many of these patients who are resistant to standard treatment. So to summarize, vitreous samples in chronic uveitis are amenable to single cell RNA-seq. This is the first time that this has been shown, because all the earlier studies were on aqueous samples. We do see that these are highly heterogeneous, and there are indeed some novel functional subsets in both acute as well as chronic inflammation. The challenges that we are facing is that no two patient samples are exactly the same. So while we are trying to demonstrate the cellular heterogeneity in the samples, we would want some element of homogeneity in the clinical phenotypes that we are seeing. Now, there is some degree of homogeneity in the clinical phenotypes, but we would want much more of it. Definitely, these need validation both on the RNA-seq, which is high resolution and small cohort, and flow cytometry, which is a low resolution but a much larger cohort. And all of these may help us in repurposing some of the existing drugs for the targeted therapy of intraocular inflammation. I would end with these two quotes by two Nobel Prize winners. One is saying that the progress in science depends on new techniques, new discoveries, and new ideas, probably in that order, which highlights the importance of today's session. And Francis Crick saying that every generation needs a new music, and probably the new music of this generation is single-cell RNA-seq. I would like to acknowledge these people, especially Sandhya and Shayantan, who are the backbone of all the work that we do. Arun is sitting in the back. He's no longer with us, but he also had a big role in the data that I showed here. And this gentleman, Professor John Forrester from the University of Aberdeen, who has been our main immunology support for the past 5 or 6 years. I've already acknowledged Pankaj before, but I should also acknowledge Santosh from 10x Genomics and Tulsi from CCMB for the wet lab experiments. All the funding agencies who supported the research that I showed here and all the other research that we do. Thank you for your attention.
Thank you so much, sir. That was a wonderful presentation. Now, moving forward, I would again request the audience to please post your questions and we'll be taking it at the end. Dr. Arivu, kindly please present your slides.
Thank you, Kushbu. Thank you, Dr. Basu. That was really insightful presentation. Is my screen visible?
Yes, ma'am.
Switching gears, I think we had a really good presentations from both the speakers, and now we are going to switch gears to just look at the technical aspects of how those data was arrived from, the methodology wise. And at Strand, just to give our capabilities, we have state-of-the-art instruments in our lab. We have the shotgun and sequencer, long read sequencer, and of course Chromium X for the single cell. And we also have high-end automation instruments to scale up and to do high throughput experiments. Dr. Basu already gave a little bit of overview of what 10x workflow entails. So as the name suggests, it all starts with the single cell suspension, which is the part of the input for the single cell preparation. And in the Chromium instrument, you get the gem emulsion beads, which is then taken forward for library construction and sequencing using 10x workflows. Currently at Strand, we have adapted 10x, but we are also working towards establishing the other pipelines that are available for single cell as well. So 10x works with droplet-based microfluidic device. So where you have this barcoded gel beads, which encapsulates every single cell, and each bead will have each gem or the gem emulsion will have a single bead and a single cell, and all the reagents that is required for sequence for RNA transcription and barcoding is all present in this bead, and for the subsequent processing, whatever is required is present in the bead. And this is the first step. This is what you get from this instrument, post which the library construction happens and sequencing and data analysis happens. This is the entire gamut that 10x workflow offers. You have this universal gene expression, which is species agnostic, and it provides the maximum versatility. And then comes the flex gene expression, where you can fix, patch your samples, and run on your own schedule. And then you have epichromatin to look at chromatin regions. So under universal gene expression, there are multiple workflows, which is 3' RNA-seq, 5' RNA-seq. Both of that can happen on single cells and single nucleus as well. Whereas the flex gene expression is a probe-based chemistry, um, and it is a flexible sampling. That's why it's called flex gene expression. You can use fixed cells, you can use tissue. FFP also is compatible with this assay. And currently, 10x even has introduced a protocol to fix blood samples and use that with this assay. And of course, for chromatin, you have to use the single nucleus and not single cells. So that will be a single nuclear RNA-seq. And in multi-ohm assay, you can also do both RNA expression and ATAC in a single experiment. So coming to 3' universal gene expression, a little bit more detail on each of these assays. So coming to the 3' gene expression, it starts with this either cells, single cell suspension or single nuclei suspension. And then the process is gem generation, followed by subsequent steps. And at the end, you have the cDNA that is from each of the cells, which has cell barcode and UMI for each transcript. And you do cleanup and you get the library and sequencing. So what to note here is that this is from fresh cells and you need cell or nuclei suspensions. It works with either fresh or frozen tissue. In case of frozen tissue, you can only do a single nuclear RNA-seq because the cells are not live anymore. And then you can use flow sorted cells, fixed human PBMCs, or organoids. This is compatible with, because it is species agnostic, it can work with any kind of species. And then comes the 5'. Both 3' and 5' are poly A capture based methods. However, like Dr. Basu's entire experiment was based on 5'. So the reason being he wanted to look at the variable regions as well at the 5' end, which is the TCR, PCR, or VDJ, all that comes under 5' region, which is why this assay is specifically suited for experiments where you're looking at immune cells and looking at VDJ regions and TCR, PCR regions. And that is where it is suited for. And like you can see that with gene expression, you can study VDJ and also CRISPR screening or cell surface protein. So all this can be done using 5' universal gene expression. This is called as feature barcoding. Each feature can be added to this main 5' gene expression. You can add a VDJ feature, you can add a CRISPR feature, or you can add a cell surface feature. Whereas in the 3' you can only add cell surface feature. This is also compatible with many different cell suspensions, and you can also use from any different species as well. Then moving on to the flex assay, which is quite, it gave the flexibility to a lot of researchers in the labs, especially in hospital settings, where a lot of people do not have the entire facility to do the experiment right away in those kind of situations, or where you want to batch the samples, where you cannot immediately do the experiment, but you want to do it on a later date. You can plan your experiment, batch your samples, and then do it on a later date. So where you can fix the tissue and store it and later on dissociate into cells and use it, or you can make the single cell suspension and fix the single cell suspension and use it on a later date. So all these flexibility is available with this assay. So here you can see that it starts with the fixed cells or fixed tissue. If it is fixed tissue, it has to be dissociated into single cells. If it is fixed cells, then you take the cells and you have these probes for RNA, which goes directly and binds the RNA inside the cells. So once the probe hybridization happens, then the process follows just like the other assays, gem generation and library preparation. Unlike the other two assays, which was 3' and 5', this is only compatible with human mouse and you can add custom probes. The mere reason being it's a probe-based chemistry. Currently, the probes are available only for human and mouse. This assay is compatible with fresh and frozen tissue, paraformaldehyde fixed and FFPE, fixed whole blood organoids and flow sorted cells as well. So all of them either starts with a fixed tissue or fixed cells. So that's where it comes as flex because of the flexibility. You can fix and store samples. The storage can be like if you are planning to do it in a week's time, then it can be stored at 4. If you're planning to do in a couple of months or 6 months down the line, then it can be stored at -80 for 12 months. And this also, unlike other methods, offers multiplexing. You can see these are color coded. It just means that prior to this generation, all these samples can be pooled. So it gives the flexibility to plex the samples. What it means that you can increase or batch up to 128 samples or even more. It reduces the cost of the experiment significantly. And you can even scale to million cells. And yeah, the main advantage is batching to a high number of samples if you have. And as your batch size increases, the cost per sample also decreases in while using flex assay. Applications of these, you went through the presentation, the brilliant presentation of both the speakers. So we already knew where all it has been applied for, like cell type identification and cellular heterogeneity, development and differentiation pathways, and to identify gene regulatory networks, of course, tumor microenvironment, immune profiling, and biomarker discovery, drug target discovery, response prediction, and much more. So then we have the ATAC-seq, which is specifically to look at open chromatin using transposase-based approach. So this is only capable, this can only be done on nucleus, and ATAC-seq can be clubbed with uh, gene expression also. So in the same experiment, you can get both RNA expression as well as ATAC sequences. It's mainly used to study gene regulation, where you identify active promoters, enhancers, and other regulatory elements driving gene expression, and also to define cell types and states based on their epigenetic profiles, and to understand how chromatin accessibility changes in different conditions under different states of cells or under different disease conditions. So to summarize, these are the different methods that are offered currently under single cell umbrella. And the first one is the 3', where you can only look at it is a full length transcript, but you capture at the 3'. And fresh life cells or nuclei can be used as a sample type. It's ideal for basic research, and the plexing is just single plex. Coming to 5' RNA, it is specifically to look at immune cells, VDJ and TCR BCR regions, and gene expression. And fresh life cells or nuclei can be used for the same. It is ideal for immunology, CAR T cells, and tumor infiltration kind of studies. Flex RNA profiling can be done from fixed samples. It can also be used with archived samples. And here you have the flexibility of plexing samples, either 4-plex, single plex, or 16-plex. That is the advantage of using this assay. And ATAC-seq, of course, to do the chromatin accessibility, tells which genes are open to be activated, and it needs fresh nuclei. gene regulation enhancer or promoter study can be done using ATAC-seq. Site-seq is to do both RNA plus surface protein expression. You get the data of RNA and few tens or hundreds of proteins data from this particular assay. Here, fresh live cells and antibody staining is done. So this antibody is done through BioLegend, which is something 10x approves of, and BioLegend antibodies are third party, but SiteSeq supports this assay using those antibodies. It can be done for immune profiling, tumor microenvironment, etc. And then we have the multiome, which is RNA plus ATAC, where you can do expression and ATAC in the same setting or in the same cell type. It's mainly used in developmental biology to understand regulatory networks, etc. With this, I would like to invite Pankaj to present on the informatics pipeline post data generation.
Yeah, thanks, Aru. So in terms of single cell analytical capabilities at the Strand, we have a well standardized pipeline to carry out all sort of the analysis. So the pipeline basically implemented an automatic filtration of the ambient RNA, the single cell and the multiple sample. It can run in the single sample or a multiple sample mode. We have more focused on the cell type annotations, considering the different approaches of the cell type annotation. That also includes the recently developed the AI/ML based approaches, and very uh, a good quality of images which can directly placed in your publications for a publication point of view. So summarizing the pipeline or the data analysis steps, we basically, the pipeline has a 4 different modules. The first module is more centered to the QC steps of the data sets which is generated after the experimental process, where it looks for the different the percentage of the mitochondrial, ribosomal, and the number of genes and others by looking the statistical method as well as generating the different QC plots. Based on this, they basically identify a cutoff threshold and removed all the unwanted cells. The second module is basically the R-based approach, where it goes through the different steps of the analysis that includes the data normalizations, scaling, PCA, cluster generations, and then also the UMAP to identify what are the different cell types present in the sample. The third and the fourth module is more exploratory, where we basically try to address the biology associated with the data set by performing the RNA velocity analysis, gene set enrichment analysis, cell stemness, intertumor heterogeneity, and the serotype analysis. And the fourth module is more centered to the specific whenever we perform the VDJ analysis or ataxic analysis, then we execute the module 4. I'll go next. So as I said, the pipeline has an inbuilt the statistical methods to identify and to filter out the unwanted cells. As you can see here in the right side, the pipeline generates such images as a part of the analysis steps. And in terms of the normalization and the batch effect correction, uh, the pipeline and the analysis method enables users to select either the log normalization based method or IC transform based method. And also, if there are the more than one sample for the integration, there's 3 different tools for the data integration is a part of the pipeline. The CCA, the basic comes with the Shura, the canonical method, then Harmony and Liger. One more added feature is that the user can select what the resolution is optimal for the cluster generations by looking the cluster plots generated as a part of the analysis method. Sorry, yeah, next. So as I said that because the cell type annotation is the most crucial and important step in the single cell overall analysis. So we basically has implemented, and the pipeline has a three different types of the cell type annotation method. One, when there is already known gene signature or the database referenced, then it uses the SC type method, where take the gene signature from the azimuth references or other databases to annotate the different clusters. Also, execute the single R, which internally resources the six different databases, well established databases for the reference point of view. It also has one added, that is the Surat level transfer. Basically, if someone already had performed the single cell experiment on the similar tissue type or and already annotated, it basically do the level transfer to annotate the newly query data set. The third is more the AI ML based approaches, where the pipeline has implemented the CGPT, GPT-4, SC Balance, SC AI, and the Cellent. All these are the AI ML based, LLM based approaches in the pipeline. So, as I said, that the most of the analysis or the key finding comes from the exploratory analysis. So the pipeline as well as the individual analysis is done for the cell type specific markers analysis, in depth cell type specific differential gene expression analysis, gene set enrichment analysis, and all sort of the analysis which is more crucial for any publication centric analysis, and the support is done by our team here at Strand. Yeah, thank you.
Thank you. Thank you, everyone.
Thank you, Dr. Arivu and Dr. Pankaj.
Thank you.
I think this is the end of all the presentations. I would be reading out the questions that are there in the chat box. Kindly please address to it. I think the first two questions are towards Dr. Arindam's presentation. The first question is, in the context of tumor heterogeneity depicted here, how do you envision distinguishing between functional heterogeneity that drives clinical outcomes versus stochastic noise in gene expression?
So, yes, it's a very good question actually, but what I didn't show the details. So, most of these stochastic variations, I mean, this variation, gene expression variations can be of multiple sources. The technical variations or the batch level variations that are taken care of using various methods in the analysis where the data is normalized. Harmony is one example, but not the only measure that is done to remove this. But at the end point, for the data that I particularly, they are all very similar. They are all very similar stage and tumor types. So they were very early stage treatment naive patients. So what we are seeing from there is basically what we feel that each of these tumor is seeded with some kind of outcome built into the gene expression program. So that unlikely to be stochastic.
Thank you, Dr. Arindam. Let's move on to the next question. If MAL1 signatures predict poor prognosis, could targeting their metabolic dependencies improve patient outcomes?
Yeah, that's actually it's again a very good comment. I would say not a question, but a comment. And that actually showcases how single cell RNA-seq kind of approaches are now bringing out these kind of infer be tested out now, and that is what it suggests, that if we address the metabolic activity in this particular cell state of the malignant cells, that might lead to an improvement in the tumor progression. So that needs to be tested out. We are actually, what I suggested is that we are at this point, after generating this amount of data, we are actually looking at the cell state in a celluloid model, actually, and then subsequently we will move up how these can be addressed. So, and the comment that is made is actually something that we are planning.
Thank you, sir. I think the next question is directed towards Dr. Somyava, but since he's not here, I would request one of you to please address it. Um, it's about I want to ask whether taking 4 or 5 individuals for single cell analysis is enough, or should we increase the number of samples, same as it is advisable for bulk RNA-seq?
So
Um,
shall I try to give some my opinion, and then maybe others can add on. I mean, continue that.
yeah,
So
definitely.
sample heterogeneity is always an issue, right? Now, when we do a bulk kind of approaches, there are certain. I don't say that there is very perfect methods, but there are certain ways to address a potential sample size and so forth. Now, in single cell RNA-seq, there are two kinds of Ns. One is, of course, N as an individual, and the second is N as in cells. So, the given the cost and other kind of limitations in single cell arts, designing single cell gardening experiments, it has to be kept in mind that these two ends. So either I can do a deep profiling of less number of individuals, or I can do shallow profiling in larger number of individuals. So it all depends on what question I'm trying to answer. But at the end, of course, two less, if you are expecting a lot of sample level heterogeneity, too less number of samples will not provide you enough heterogeneity to start with.
Yeah, and just addition of this that the 4 more samples is added. So now the total is total 8 patient data is included in this analysis to consider. And the good thing is that both the blood data as well the PBMC data as well as the uveitis data, ocular inflammation data is the part of the analysis. So to have a clear picture of the normal blood as well as the infected conditions.
Thank you, sir. I request the audience to please raise your hands or put your queries in the chat box. We'll address it. Until then, I would like to ask the panelists, do you all have any questions for each other's presentation?
Yeah, just I have one questions to Dr. Arindam. So, as you have identified in the analysis the double negative cells, what was the criteria to identify more specifically in the single cell experiment? The CD8 populations are well characterized with the CD8A or B expression, but it is noted that the CD4 expression more particularly is very less in although the cells are the CD4, but the CD4 itself expression is very low.
Yes, I
So,
agree the CD4 levels of expression are poor. And that's why what we did was we subsequently, using the tumor cells, we also did flow experiments and ICC, where we actually showed that actually we are getting cells which are CD4 negative, CD8 negative, and PLCG2 high.
okay.
So additional evidence is required in such kind of cases, and here also I did that.
Yeah, okay.
Dr. Swaraj, Dr. Arivu, any questions from your end?
Dr. Mehta, just for the audience educational purposes, if you could also tell them, like you were saying that you are planning other experiments to, um, validate the single cell. So, if you could explain what those experiments entail, so that would give audience a workflow of what should they plan if they have to do an entire experimental.
Okay, so these are like standard, models in biology. So one is, of course, Dr. Basu pointed out about the CD4, CD8 negative. Use flow and ICC to show that these are not artifacts of analysis, but these are actually true cells which are present in the tumor. And then what we did, what we are right now doing is taking those forward. We are inducing higher expression of, so we are isolating double negative T cells, inducing high expression of PLCG2, and then trying to see how the cells behave, and then how do they interact with malignant cells. in a co-culture system. So these are some things that we are working out. Also, like someone made that insightful comment that for this different malignant cell types, we are trying to generate a celluloid model for this MAL1, MAL2, MAL3, MAL4, and then see if we shut down the expression of these signatures, or how does the cellular phenotype change. So these are experiments right now we are
Okay,
at some example.
thank you.
Thank you, Dr. Arindam. Are there any more questions from the audience's end? Okay. Yeah, no problem. You can also post it on to our mail IDs, and we will try to route it back to you. I would request Dr. Swaraj to please share the vote of thanks. So you're on mute.
So, yeah, firstly, I'd like to thank Dr. Arindam, Dr. Basu, Pankaj, Arivu for putting forth these different aspects. Specifically, when we have these kind of talks, personally from my side, when I hear them, what heartens me is that we are coming close towards translation, because till now single cell experiments have been used purely from the perspective of exploration, purely from the also from the perspective of digitization of the currency we call mRNA. So bulk had a certain level of flexibility, and then we had single cell. And now when we see these specific cases of oral cancer, other cases, we can clearly understand that we are coming close to translation. And translation is where understanding the biomarkers, understanding the clinical translation of it, understanding how soon we are in a position that if I am running a clinical trial of 10,000 patients, I can postulate something in the lines of PBMCs and run it across all the patients and understand what those PBMCs are telling, and whether their signatures are something very close to the tumor biopsy. So that's something which I am taking forward. Personally, I would say that the presentations have been extremely enlightening for me. And I also thank especially Dr. Arindam to have given it in such a robust manner that it simplifies the entire context. So basically, when Khushbu said that, is there any question? I particularly didn't have any question, because most of the things which I would have planned to ask were covered in the deck itself. And thanks Arivu, thanks Pankaj. We are doing wonderful things in Strand from the single cell perspective. We think that we are going to go places, specifically with the volume of efforts we are putting on our pipelines and on our methodological robustness as well. I keep sticking to the point that at Strand, we always come with the motto that we do computational biology, but it has to be always backed by science. We build our statistical methods, pipelines, but it is robustly backed by science. Sometimes it takes a bit more time than expected, but that is the norm when we want to be very sure of the QC and the processes. With this, I'd like to thank Khushbu for organizing this webinar, and thank you everyone. Thanks the audience for being here.
Thank you, everyone. Thank you, Dr. Swaraj. And I thank all the audience for staying throughout the webinar. And also Dr. Arindam, thank you so much for accepting our invite. And Dr. Soumyava Basu as well. I thank Dr. Arivu and Dr. Pankaj as well for coming and sharing this insightful presentation. Audience, I have given a mail ID in the chat box. If you all have any other questions related to this webinar later, you can please reach out to us. We will share the answers with you as soon as possible. And thank you, everyone. Thank you so much for making this webinar a success. Thank you,
Thank
Dr.
you.
Arindam.
Thank you, everyone.
Thank you. Thank you, Dr. Adam.
Thank you.
Thank you, Arivo. Thank you, Suraj.
Bye.
Bye.